This is Dr. Charles Parker, and you're listening to Core Brain Journal. It's a place where I connect both fresh discoveries and interesting different perspectives from advanced mind science with the realities of real people and everyday life down on Main Street. Well, welcome, folks. Here we are one more time at corebrainjournal.com, and I'm really pleased. This is going to be a very interesting conversation for me because every time I'm with Dr. Bill Walsh, I learn many things that can be applied directly to my practice every day. And I think what he's done is he's just contributed a considerable amount to how psychiatry is changing in the world today. Bill, thanks so much for joining us. I appreciate it.
Well, it's my pleasure, Charles.
So here's the thing. What I'm going to do is read a brief intro, and then we're going to get right down to talking about methylation and what does methylation even mean? Why would we be concerned about methylation when it doesn't seem to be on the radar of so many people?
Yeah.
So Bill Walsh is president of the nonprofit Walsh Research Institute near Chicago. He directs doctor training programs in the U.S., Australia, Norway, Ireland. He just got back, he told me, from Japan. And he's a very busy guy with training. During his more than 30 years as a research scientist and engineer, Dr. Walsh developed a science-based nutrient system that has helped thousands of patients that are challenged by behavioral disorders. Get this list: depression, anxiety, schizophrenia, ADHD, autism, and Alzheimer's disease, and is used by doctors throughout the world. Dr. Walsh has researched biomedical details for individuals previously considered as untreatable. It's a magic word for me, is treatment failure. From personality disorders to others insufficiently treatable with modern psychopharmacology. I like the way you said that, Bill, because we can get some of it right, but if we don't get it right, it's not right. So from that, we're going to talk about nutrient imbalances that alter key levels of neurotransmitter, key neurotransmitter levels, disrupt gene expression of proteins and enzymes, and cripple the body's protection against environmental toxins. His book, which I'll have listed in the show notes, Nutrient Power, the most recent edition is 2014, describes his findings in detail, and it really puts in, operational perspective advanced nutrient therapies which can be used every day in anybody's practice. So enough of that. Bill, thanks again for coming on board. I appreciate it.
I'm happy to be here.
So the thing we want to start with on methylation, it's a term that people really just aren't familiar with. My particular group of listeners, we have some that are familiar with it. And of course, there's more and more talk about methylation because people are looking at it from different perspectives. But let's talk a little bit. Why would we even be concerned about methylation? What does it do? What's the issue?
Well, first of all, methyl is carbon with a few hydrogen atoms, and methylation is essential to more than 80 really important reactions in the body. And what's another thing that's really not well known is that while a baby is developing in the womb, during that first 3 months, the body is almost miraculously deciding how to provide exactly the right chemicals for every part of the body. Because we have the same DNA in every cell in our body, but every part of our body needs different chemicals, different enzymes, different proteins. And all day, every day, our DNA is supervising and orchestrating the chemicals that are in our liver, our skin, or whatever, and that's all set up before birth. So under methylation or over methylation, our methylation characteristics are innate. They're fixed before we're born, and if a person happens to be undermethylated, there are chemical differences that will occur in this person's body, and it will affect their behavior, their symptoms, their traits, and we know what those are. So we're able now with lab work and with specialized lab tests together with a good medical history, learning everything about a person's symptoms and traits, we can do a very good job of identifying or diagnosing whether a person has normal methylation, and that's 70% of the population, whether they're undermethylated, which I am, and I think you too are, Charles. I think
Yeah,
we're
that's
undermethylated,
true.
which is not always a bad thing. And that's 22%. And then 8% are overmethylated. And now I've studied more than 30,000 patients directly with the assistance of doctor colleagues. And we've determined the methylation status for all 30,000. And anyway, we know a lot about that, but what's really important is that it's for people with a mental challenge, whether it's depression or anxiety or schizophrenia or ADHD or even a behavior disorder, it's really important to know what a person's methylation status is. Because if a person has, for example, suicidal depression, and if they happen to be undermethylated, if we give them methylation treatment, most of them get a lot better and may not need medication anymore. And the same thing is true of other conditions. So methylation is critically important, and it has everything to do with specific reactions in the body. But then beyond that, in the last, I'd say 5 or 10 years, we've learned a lot from the emerging science of epigenetics. And epigenetics is really dominated probably by methylation more than anything else. Epigenetics, for those who may not be familiar with it, is basically the system by which the body regulates what genes do. Epigenetics regulates which genes are expressed and which ones are silenced in every part of the body, every cell line, or every different organ in the body. And so it has extreme importance. And what we didn't know up until recently, we didn't realize that nutrient therapy and drug therapy can directly change the epigenetics. And for example, one of the most important factors in mental health is serotonin reuptake or dopamine reuptake. And these are the dominant factors that determine how much serotonin neurotransmission you have. It's not how much serotonin's in the brain, it's what the reuptake is. Reuptake basically is the rate at which serotonin, once it's in the synapse, the rate at which it goes back to that original brain neuron. And that's reuptake. And about 25 years ago, the medical researchers, especially pharmaceutical researchers, learned something very important. Up until that time, they spent a lot of effort with drugs, or even nutrients, trying to increase serotonin concentrations in the brain. And 25 years ago, actually 30 years ago, about 1985, was the time they discovered, no, it's really reuptake that dominates. Reuptake is maybe a 95% factor, and the amount of serotonin is only 5%. Now, we were able to do some good with L-tryptophan and a few other nutrients that can enhance the amount of serotonin. And medical people, the pharmaceutical they used to use were MAO inhibitors, which simply increase the amount of serotonin in the synapse. But they went away from all of that in 1985 when they found out that reuptake was everything. So what's new is in the last 5 years, we now know what nutrients can enhance reuptake or which nutrients can inhibit reuptake. So we now with nutrients can actually do the same thing that Prozac and Paxil and Zoloft can do. We can also choreograph with nutrient therapy neurotransmission of other neurotransmitters. We know how to impact GABA, the NMDA receptor, dopamine, norepinephrine. It's wonderful. This is all very new, and it's very exciting.
See, Bill, this is very exciting for our listeners because to kind of wrap up a little bit what you just said for the folks who are really more on the lay level, what happens, what I see in my office so often is people who are treatment failure, and I, knowing good psychopharmacology, go in there and do what I think is right for them based on their superficial appearances and their clinical condition, the way they look on the outside. And some of them, I can just give them medication, give them medication, give them medication, and they're treatment failure no matter what you do. To put it another way, there just don't appear to be any neurotransmitter chickens in the chicken ranch. There's nothing there. On the other hand, we have some people that come in, they're suffering enormously. They don't know what to do, and you give them just a little bit of something and they blow out the top. They just have all kinds of side effects to medication. So they're a treatment failure on the point that a little bit causes harm, or they're a treatment failure on the point that nothing is working whatsoever. And somewhere in there is what the methylation is all about.
Well, methylation is important in about 70% of all people with a mental problem. Sometimes something else is wrong. Once in a while, it could be metal metabolism, like a copper imbalance or a zinc imbalance. It might be a pyrrole disorder for maybe 15%. Or for a small number, it can be toxic overloads, too many toxic metals or whatever. But about 65 to 70% of the time, methylation is directly involved. And it's individual. Sometimes it's over-methylation, but more often it's under-methylation. And it's really important to find out. And now we're able to find out.
See, that's a key point. I mean, measurement is what it's all about. It's something we've been talking about. I'm so enthusiastic about measurement. I'm so appreciative meeting you because with measurement, we've been able to see people turn around much more effectively and more completely. Yeah, we were able to do something before, but when we get in there and actually measure, it's a whole different thing. Now, let's start by really talking, because some of the people know something about methylation, and they've heard about this whole controversy about MTHFR. It's surprising to me, Bill, right out here on the streets, as it were, how many people are now doing MTHFR and genetic testing. And so if we could talk a little bit about that, some of our listeners are, I'm sure, going to be very interested in hearing what your thoughts are about MTHFR as it relates to methylation.
MTHFR is part of a very complex, what we call a one-carbon cycle, or the methylation cycle. Others call it the SAM cycle, but basically it's a cycle that enables us to recycle methyl. We get our methylation from diet, from the amino acid methionine, which is present in every bite of protein that you take. And the methionine then has to convert to something we call SAMe, because it's hard to say S-acenosyl adenylyl methionine, so we call it SAMe. And SAMe is a fairly unstable molecule that readily gives up its methyl, and it's the primary methyl donor throughout the body, throughout the brain, and even with respect to our DNA. It's what donates methyl. Now, once the methyl is used, once it is provided by this methylation cycle, it has to be regenerated. It has to be controlled and regulated. It's regulated by a very complex cycle that I'm sure some of your listeners know everything about. Now, there are about 7 or 8 important enzymes that enable each one of these reactions around that cycle. It turns out that the most dominant, in general, the rate-limiting enzyme happens to be the one called MTHFR, and that's an enzyme that creates something called methylfolate, which is very good at reacting with homocysteine together with B12, and so what people have done with genetic testing is they focused on SNPs, or which are called single nucleotide polymorphisms, which really are mutations. MTHFR is a huge molecule. It's genetically expressed. It has more than 500 amino acids, so it's a big guy. It's got a lot. And a SNP, basically, a mutation, is simply one of those 500 aminos out of place. Where maybe you were supposed to have a methionine, you might have a glycine or something like that. And by the way, all human beings have hundreds and usually thousands of mutations in SNPs. We all are loaded with SNPs and mutations. Most human beings have at least 10 or 15 mutations in their methylation cycle, and more than 10 in MTHFR itself. Now, since these MTHFR is such a large molecule, it depends on the location of the misplaced protein, the misplaced amino. And we now know that the 677T location, and it's really counting just 677 aminos from some starting point. And there's another one that's called 1298. That's number 1298 along, measured along this list. And by the way, it goes to 5,000. These are the two that we know are the most impactful on a person's health. And impactful because if you've got a weakness in those areas of that enzyme, you tend to be undermethylated. It tends to reduce the ability to provide enough methyl for the body, in short. Now, there's a lot of people doing genetic testing because I think genetic testing is wonderful. I love it. It's a great analysis. It's high tech. It's science. However, there's a lot of people who are mistakenly using this information as a diagnosis for methylation. And unfortunately, it's not very efficient. The problem is that, yes, if you have an MTHFR 677, what they call homozygous, where both copies of your DNA have the same mistake, yes, that will weaken methylation. And if you want to improve your methylation, which would be nice to do, the best way to do that is with methyl B12, some form of folate, folic acid, folinic acid, or even better, methylfolate together with B12. And if a person simply does that, their methylation will improve. The problem involves mental illnesses. And quite a few people have low serotonin activity. And what we didn't know until recently is that folates, even methylfolate, all forms of folate, lower serotonin activity. And so if you take a person who's depressed, say they're suicidally depressed, and they're trying to take Paxil or Zoloft, and the side effects are not tolerable, and they want to increase their serotonin activity, and they're undermethylated, as many of them are. If they were to take methylfolate, their methylation would improve, and the patient would get worse. And the reason the patient would get worse is that all the folates are powerful serotonin reuptake promoters. And what you need are serotonin reuptake inhibitors. You want to increase serotonin activity for these people. So while methylation, you can many times improve methylation in somebody who is low undermethylated, and yet the patient will react horribly to the treatment. And that's what I think the confusion is, because all these nutritional biochemists are correct. If your goal is just to improve methylation, then folates and B12 is usually the best way to do it. It's considered a gold standard way to improve methylation. The exception to the rule are people with mental health problems involving low activity at either serotonin receptors or dopamine receptors, and that's a lot of people.
So let's say that, let's freeze frame that again, because what's going on, we're talking about transporter proteins on the presynaptic nerve. And those transporter proteins are either more or less active based on this methylation process. So when you say reuptake, it means it pulls it right back up, so it's not in the synapse and it's not working effectively. It's gone. It's pulled out of action.
You know, one of the amazing things about neurotransmission is that when a cell fires, say a serotonin cell fires, the serotonin, these vesicles that are at the presynaptic membrane, they get shot and injected in the synapse, where they can do some real good in communicating with other cells. However, what's almost a miracle is how rapidly the serotonin goes back it goes right back into that original cell. The passageways for the return are these, they're called reuptake proteins or transporters. And the one for serotonin is called SERT, S-E-R-T is the name we give to the serotonin transporters. Well, SERT is genetically expressed. Now, if a person takes Prozac or Paxil or Cerazone or any of the SSRIs, they quickly go to those very transport proteins and they sort of disable them and weaken them. And that's how they inhibit serotonin reuptake. So the serotonin stays in the synapse longer, and the person gets better if they have low serotonin activity. What nutrient therapy can do is a bit different. What we can affect is genetic expression of SERT, of this transporter protein. We can cause the body to gradually, over a month or two, decrease the number of these transport proteins. So we're not disabling them, we're reducing the population where it counts, and that inhibits reuptake. So we're doing the same thing, only ours is done in a natural way with no side effects. Absolutely no side effects.
So that's the key to point. Now, you broke up there a little bit, Bill, so I'm going to just say what you said again just to make sure everybody's on it. but it has to do with those reuptake proteins, the transporter proteins. What you were saying is that genetically what happens is if you use the correct nutrients, you can modify those transporter proteins. That's the bottom line. If you diminish their effectiveness, then what happens is the serotonin remains more active in the synapse. And if they're flipping out the other way, if they're way overactive, you can turn them off and help them actually help act as a, it won't keep them so much in the synapse, but pull them on out of the synapse because they're kind of synaptically overloaded.
Yeah, exactly. You want to try to trap the serotonin there. To me, it's like a ping pong bouncing back and forth, trying to activate other cells. And the longer that's there, the longer the residence time, the more activity you're going to get, and the better the patient will be. The problem is that what people have only understood recently, and a lot of people still don't realize this, that folates act epigenetically to greatly increase the population of those transporters. They just load up your brain cell with these transporters so that the serotonin disappears too fast.
Now say that again, Bill, because you broke up right there. Say that last thing you said. What is it that goes in? Because I didn't hear it myself.
What folates do.
Folates, okay.
Folates.
Yep.
They affect epigenetics very powerfully, and they tend to increase the rate of genetic expression of these SERT transporters. And so you tend to load up your original neuron, called a presynaptic neuron, and loads it up with these proteins, and so the serotonin disappears quickly.
Pulls them right out.
Exactly what you don't want. So you have weakened serotonin activity, and that's very important for many people with depression, OCD, schizophrenia, bipolar disorder, even behavior disorders. So if we find somebody with that, so we find an undermethylated person who has a low serotonin problem, low serotonin activity, we'd love to give them methylfolate to increase their methylation. The only problem is they would get worse because it would damage the major problem, which is serotonin neurotransmission.
Now, Bill, we've seen that clinically. This is one of the important things that I learned from you years ago when we first met out there in Palm Springs at the first meeting I met you. And then I came up and had that great time with you up in Chicago, and it was really transformational for me because that particular thing, I was using Deplin, but at times I was using Deplin with the idea that I was going to be helpful with this L-methyl, and people would get worse very quickly, and this is the reason they would get worse.
There was these people, the people with low serotonin activity, that were getting worse. And the Deplin can be wonderfully effective for many people. The exception to the rule are people who suffer from low serotonin activity.
So what, to say it again, undermethylation meant that they actually had a paucity of serotonin in the first place, and putting the folate on just pulled actually more of it out. So they and what happened is they had a paucity of serotonin. Go ahead, you were going to say something.
Well, not so much a paucity of the serotonin, but the serotonin that is ejected into the synapse when a cell fires disappears too fast.
I got you.
It's
okay.
Not the amount, it's more the rate at which how long it stays there.
Thank you.
That dominates everything.
See, that's a good point. That's really a point. That whole dynamic flow is an excellent point. I appreciate your making that, because I was thinking a little more structurally and a little more statically. And it's really a dynamic process. It's boom, and it turns around. It just doesn't turn. It's a rate step.
What's amazing is that there are brain cells and neurons that can fire 6 or 10 times a minute. Some of them can do this several times a second. So that reuptake can be really, really fast.
Now that is so interesting. So that MTHFR is only one piece of a very complex picture. There are many, many things. So then what is the resolution that you came up with, Bill, about how to measure, not so much on a specific qualitative, but on a more quantitative level, the larger picture of what's going on in the synapse?
Well, after working with 30,000 patients and studying the biochemistry, but really learning from the patients, we've learned that one thing that is really helpful is that people who are undermethylated people who have low serotonin activity are born with characteristics that are quite distinctive. For example, they tend to be perfectionistic. They tend to be competitive. They tend to be they tend to be a bit OCD. 75% have seasonal allergies. They tend to have a low pain threshold. We have a list of 20 or 30 characteristics that are associated with undermethylation. And we also have seen enough overmethylated people. We know what their characteristics are. And so we can do a pretty good job of diagnosing methylation with a really great medical history. But of course, we like to do lab work to verify it. Now, the problem is that a problem with methylation is not just undermethylation. You can also have overmethylation, which a lot of nutritionists have not really focused on. And like I said, 8% of the population is overmethylated, 22% undermethylated. Now, how can a person become overmethylated? Let's say the methylation cycle's working fairly well and generating enough SAMe to do all these 80 important reactions and also regulate epigenetics at the DNA level. Let's say you've got that's working pretty well. The problem is, what happens if you can't utilize the SAMe? More than half of the methyl that the methylation cycle is creating and synthesizing is used for one single reaction, and that's the synthesis of creatine. About between 40 and 70% of all your methylation goes into one reaction. Well, that reaction also has enzymes. There's two steps to the reaction. There are two enzymes that also have SNP mutations and can be very weakened. So it's possible to have a glut of SAMe. If your one carbon cycle is pumping out enough SAMe, but if you're not using it, you've got all this extra SAMe floating around, and you tend to be overmethylated. This really affects genetic expression, and these are the people who tend to have, for example, really high norepinephrine and very high dopamine activity, and it's probably 70 or 80% of people with panic disorders or anxiety disorders. And it's also related to many people with schizophrenia. Classic paranoid schizophrenia, almost all of them have this situation where they're over-methylated and they suffer from extraordinarily high levels of dopamine. Now, people have known that in the schizophrenia field for 30 years. For 30 years, we've had the dopamine theory of schizophrenia. As a psychiatrist. I'm sure you learned that in medical school. And so most of the therapies have been aimed at reducing dopamine neurotransmission. Well, the reason the reason they have too much dopamine activity is they have a folate deficiency. These people need folate. This is the opposite of undermethylation. These people need folate to shut off you want to increase these, in this case, the dopamine transporters. There are also dopamine transporters called DAT or DAT. And so the therapy for these people, even though they're overmethylated, strangely enough, is to give them folates. And the reason is epigenetics. And it's wonderful to know that. Now, this was a mystery for about 30 years, and it bothered the heck out of me. It bothered the great Carl Pfeiffer. It bothered the great Abram Hoffer. We had studied many, many people, and we realized that undermethylated people were made worse by folates, and overmethylated people got better on folates, and it seemed totally counterintuitive. And the reason is we know folates are great at methylating, but we were getting the opposite effect. And thank God, about 6 years ago, we learned why. The mystery was solved. It was solved by the emerging science of epigenetics. And now we know that folates have a dramatic, powerful effect on reuptake of neurotransmitters. And it overwhelms and the effects of just fixing methylation. It's so good to know that. I can sleep better at night.
It is. Hey, I do too. I'm telling you. You feel like you can. It's actually. I was amazed when we were sitting down there in outside of Chicago at the meeting. You know, when all of us were in the room, we may have had 25 psychiatrists in a room, and a person would say a couple words, and Bill Walsh would say, here's what's going on, because you've heard it so often. Yeah.
A lot of people are trying hard to diagnose methylation with genetic testing. And if they find a serious weakening in MTHFR, they automatically assume this person needs methylfolate. And they'll also point out, the people who are really solid and great at biochemistry and study this, they point out that if you're undermethylated, it diminishes the synthesis of serotonin.
Mm-hmm.
And they're right. Their biochemistry is correct. The problem is their perspective is wrong. That happens to be a 5% effect, whereas the epigenetic effect is 95% and overwhelms the small benefit.
That is a very interesting point, isn't it? I mean, that really, that's where the rubber meets the road, definitely.
I have an unfair advantage over all these other people, and it's that I've had 30,000 patients, and I know the right answer, and only recently understood why. But it's if you just looked at biochemistry alone, it would be pretty hard to work out what would be the best thing to do for a patient. But it really helps to have thousands and thousands of patients, and it really helps to. We now have, I mentioned that there are methylation SNPs, weaknesses in the formation and the regulation of methyl, the SAM cycle, the methylation cycle, and also the SNPs that can cause overmethylation. And I call this a tug of war. And the question is, which is dominant? What wins the war? The tendency for overmethylation or the genetic tendency for undermethylation? Now, the genetic tests I've never seen one that measured the enzymes that cause overmethylation.
Mm-hmm.
Eventually,
yes.
somebody will eventually work that out. And I think eventually genetic testing will really help us diagnose methylation, not so much methylation, but the neurotransmission aspect of methylation. And I'm looking forward to that. It'll take a while. Fortunately, we have 2 lab tests that can tell us who won the war. So along with our medical history, that gives us already a pretty good idea of who's undermethylated and overmethylated and who has a serotonin or a dopamine problem. We have 2 lab tests that are very good markers to find out who won the war, this tug of war between the various SNPs, the various mutations. And one we've been using for 35 years. It was started by the great Carl Pfeiffer and Abram Hoffer together, and that is whole blood histamine. Now, the reason that is a good marker for methylation is that histamine and methyl, SAMe, are throughout the body. We call them ubiquitous. They're everywhere, in every part of the body. They're right next to your DNA. They're everywhere. And they're in pretty high concentrations. Well, histamine is regulated by methyl. It's the way it's regulated in the body, not in the GI tract, but everywhere else, and in your brain. Your histamine is, they say, metabolized by methylation, and that's the way it's regulated. So it turns out that if a person has high histamine, that means they don't have enough methylation to do the job. So high histamine is a marker for undermethylation. Low histamine is a marker for overmethylation. Now, recently, about 10 years ago, some really terrific methylation scientists at universities, for example, Dr. Jill James and Dr. Richard Deff and others who've now been really homing in on this, they have developed a test called a SAMe SAH ratio test. Well, that's a mouthful, but basically they've developed a laboratory system where they can measure a number of the important factors in that methylation cycle, and they now have what is considered a gold standard test for methylation, and that a blood test. These are both blood tests, which is great, and the SAMe test works also, although it's somewhat more difficult for people to evaluate. I think not a week goes by and I have a doctor who we've trained who started using the test and says, hey, I can't figure out how to interpret this. But it's a wonderful test once they understand it. So we've got 2 blood tests that can confirm the diagnosis and gives us a roadmap for treatment. Tells us exactly what to do to help these people.
So, Bill, let me ask you to elaborate on this a little bit for the one that most people have some familiarity with, which is a whole blood histamine, because people will be saying, Well, can I just do a blood histamine? What's the difference between a whole blood histamine? Two questions. What's the difference between a whole blood histamine and a blood histamine, number one? And number two, what kind of preparation should a patient have before they get their histamine level that would be render it accurate as opposed to being somewhat inaccurate?
Well, it turns out that in our blood, very little histamines in the liquids, the plasma and the serum. There's very, very low levels of histamine. Almost all of it is in our blood cells, in basophils, and so you have to use whole blood, or you could use packed cells. But all the data, all the reference normals are based on whole blood histamine, which is basically you take a blood sample and you don't spin it down. What you do to get good results is you simply rock it back gently for about 10 or 15 seconds and freeze it, and then do analysis of the entire sample. You don't centrifuge it and spin it down, and it works beautifully. The problem is that if a person is taking antihistamines just before the blood draw, that can knock your histamine level down and can give you a false result. Mm-hmm. So that's an issue with that whole blood histamine test. So if a person is taking a powerful antihistamine, if without difficulty they can stop it for a couple of weeks, it would be a great idea and would help add to the validity of the test. One nice advantage of the other test, the recent test that measures SAMe itself, and then the product of that reaction, which is S-adenosyl homocysteine, which is called SAH, that test is not affected by antihistamines. And that's the great advantage of that particular test.
Oh, that's interesting.
So if a person has, let's say a person's on Celexa, and they want to know their methylation, Celexa happens to be a really powerful antihistamine. And most people taking it, if they're helped by this medication, by this antidepressant, they probably aren't comfortable with stopping it for a few days, or certainly not for a week or two. So they're great candidates to take the other more complicated test. But for other people, if they're not taking Benadryl, or probably the people who really can't use the whole blood histamine test are people who are taking allergy treatments using antigens. Mm-hmm. Injected or oral antigens. We do test them for whole blood histamine, and if they're high or even normal, we know they're really high, because they would have been higher if they hadn't been doing that. So there's a lot of art to this evaluation. But now we have two lab tests that apply to everybody, one or the other. And so we are able to do that. But we have to be, for people where we suspect methylation is a problem, if we want to use the more common and the more, I guess it's more reliable in many ways, the whole blood histamine, we'd like them to be free of antihistamines to the degree that they can, for as long as they can.
Now, what is it doctor's data that has the other one that you were talking about, the SAMe SAH?
Yeah, doctor's data was involved with testing for a lot of autistic kids, mm-hmm. Tens of thousands of them, I believe. And methylation, we know, is really important for autism. In fact, I was the first person in the world to discover that methylation was a problem in autism, and that more than 95% of all autistics, all people challenged by autism, are undermethylated. Nobody knew that until 1989, and I had a huge database for autism, and we discovered that. Well, so as Dr. Deff and Dr. Jill James and others got into this, they confirmed it. And now we know that undermethylation is a key factor in autism. And in fact, I personally believe it's the reason why people are autistic. I think it's the primary vulnerability to autism is what happens in the womb as that little fetus is growing, and these methylation bookmarks are regulating which genes are expressed and which ones are not in every part of the body. And anyway, the ability to identify methylation characteristics is really important, and it can affect so much of us. Now, it's not always a bad thing to be undermethylated. Most doctors are undermethylated. Mm-hmm. I've tested some of the greatest athletes in the world, heavyweight champion boxers, NBA players, football players, Olympic athletes. Most great athletes are undermethylated. They're driven, they're competitive, they're perfectionistic. However, along with the good side, some people that are undermethylated suffer from depression, anxiety, in some cases schizophrenia, and in fact behavior disorder. People, for example, children who are oppositional and defiant, more than 90% of them are undermethylated. And if you give them methylation treatment, a lot of that fades away. So the interesting thing is that most people who are undermethylated don't need any therapy. They really don't need to do anything. But if they have a mental problem, then I think they should consider a methylation treatment, because it might really help them.
Oh yeah, we've seen that, Bill, and we're so thankful that you were on top of it, because we've seen it repeatedly. Let me ask you this question, because different people have different approaches to this. I know some of the people you worked with have a different approach to this particular question. When a person gets tested and you have a baseline, we know that undermethylation, for example, takes a longer time to turn around. So what's your recommendation for people regarding retesting and staying on top of that specific set of data?
Well, really, with respect to methylation testing, the important thing is the diagnosis. Innate inborn methylation tendencies.
Okay, hold on.
And I believe if you
broke up a little bit there, Bill, right there. Say that last thing you said again, please.
Our goal is to identify a person's innate inborn methylation tendency.
Gotcha.
It's inherited, it's genetic.
Yes.
We are born either with undermethylation tendencies or not. And it all happens usually in the first 2 to 3 months of that little fetus in the womb developing. That's when the methylation really affects our DNA permanently with respect to, and sort of defines to a large extent what we're going to be like in terms of our traits and our personalities, and also our possible illnesses. So once a person is clearly diagnosed with overmethylation, let's say they test with really low blood histamine.
Yep.
And then they get treated for that, and they get better, and then maybe you retest them again and find they're low again. I don't think they should be tested again. You've got your diagnosis. And the same thing is if you get a person who's high histamine and they do well on a undermethylation therapy.
Mm-hmm.
I think I never am happy unless I get a second lab result, because the best of all labs can occasionally make a mistake. But if I can confirm a person is a high histamine person, you don't really need to do it again. And the reason is there's no endpoint with histamine. For example, if we have a person who's zinc deficient, after we treat them, we expect their zinc to be normal and their blood levels to be normal. If we get a person with a pyrrole disorder and we treat it, we expect their pyrroles to normalize. The exception is histamine. If we take a person with a histamine disorder and treat it, and they get, even if they get completely better, their histamine may still be abnormal. And we don't care. It's not an endpoint. It's a marker for diagnosis, not a measure of how they're doing.
So to just say that a different way, what you're really looking at is you start with the baseline on the biomedical review, but in the end, the issue is their clinical condition. And if their clinical condition is good, you're done with it.
We have many times that most mental illnesses we're fighting genetics. And it has to do with the genetics that are established, the gene expressions that are established before we're born and are permanent.
Well, apropos of that point, are you saying then, I'm really trying to understand what you're saying a little bit more, are you saying that they're very likely to remain undermethylated to some degree but may not show it clinically, and you would need to watch that over time?
Yeah, I used to work with testing basketball players, and for example, I'm sure Michael Jordan is one of the most undermethylated people in the world, and he is just so competitive. And I don't think you don't want to change him if you're a basketball. If he's on your team, you want to leave just the way he is. I know obsessive compulsive, very, very talented and successful people. Most CEOs are undermethylated. Most doctors are undermethylated. Most scientists, not all, but most scientists are undermethylated. Most great athletes are undermethylated. It's only those who develop a mental issue that need the treatment.
So what you're saying, you start with that biomedical evaluation and you watch them, and if the clinical condition requires it, you go in the same direction. You'll very likely be right on either the over or under methylation because that's the way they were originally.
Yeah, and I just want to add, it's not just mental effects. Way back early, about 35 years ago, the great Carl Pfeiffer invited me to bring a number of criminals to him. And I started bringing very violent people fresh out of prison. And when I had a group of sociopaths, he forced me to go through the lab work. I don't like needles, but he said if I'm asking somebody else to do this, I should be willing to do it myself. They took about a quart of my blood, analyzed it, and he told me I was a high histamine, undermethylated person. And he gave me a treatment program for it. And I did it. And what happened to me, I don't believe I've ever been depressed. I do tend to have some OCD tendencies, but I used to have migraine headaches that one doctor once called a histamine headache. And every month I would have a really nasty migraine headache. I also had seasonal allergies that were awful. And every August and September in Illinois, I wanted to go somewhere else. And often I would take a vacation for 3 days, someplace where there was no ragweed. And since I took those Pfeiffer's treatment, I have not had a headache, and I have not had problem with allergies, unless once in a while I don't do a good job of complying with the nutrients, and it comes back a bit. So there are other things you can gain by fixing methylation.
So are you saying you stay with them a little bit? You stay a little bit with SAMe just to keep the whole thing even along the road?
I take it every day. I need 400 milligrams of SAMe a day, and that really works for me. He also found out I was zinc deficient. Now, most people get all the zinc they need from their diet. Some people, because of genetics, are very zinc deficient, which affects a couple of important neurotransmitters. And it also has a lot to do with your health, as you know.
Yes.
And your immune function. Well, he found out that I'm extraordinarily zinc deficient because of genetics. Over the years, I've learned I need 100 milligrams of zinc a day just to get my blood level just where it needs to be. I think that's why I'm still alive. I probably would have died of something if I didn't know this. So everything has to do with biochemical individuality. And when a person has a serious problem, like a woman who contacted me yesterday about her daughter who had some sort of a mental breakdown, if a person has that, or even a person with some people have terrible temper and rages, that also has a lot to do with biochemical individuality, and those people are especially easy to help. It has a lot to do with their inborn genetics, not quite so much with how they were raised and their nurturing, or how they or their life experiences. Very often, they may be born with a tendency to be exactly like that, and it can be corrected.
See, that's very interesting, and it's very helpful. So your custom would be to repeat a methylation evaluation 3 to 6 months, 3 months? What would you think it would be?
I would wait at least 3 or 4 months. Yes, I would. And if I got the same lab result and it corresponded with what I know about the patient from the medical history, then I think this person would want to have methylation nutrients probably the rest of their lives. Now, let's say you were to try this, which you may or may not need it at all, but let's say you gave it a try, and if nothing happened, I would suggest you stop doing it. But if you took it and maybe you felt better in some way, or with me, it helps me with obsessive compulsive tendencies. I mean, I'm very obsessive compulsive. When I wake up in the morning, I have this urge to do a Sudoku puzzle.
It's a handicap of being really smart, Bill.
Well, I don't know, but it's great when I dive into, if you can channel it into a career or into an athletic, it can be good. I'm very nasty on a racquetball court. I won the Illinois state championship for racquetball out of competing with 80,000 people.
Oh my gosh! Oh, good for you.
I'm very nasty, competitive, only on a racquetball court. But I can't help it. I don't think it's wonderful at all. I'd rather be a calmer, nicer person all the time.
That's so funny.
I mentioned that over-methylated people it's not a bad thing if you're over-methylated people. They are about the nicest people in the world as a group. a lot of over-methylated people don't really have a serious problem. They make the best neighbors. They're caring, they're considerate, they volunteer for things. They're not competitive at all. They often care more about other people than themselves. And so a lot of them are nurses. And if I could pick my next door neighbor, I'd want an over-methylated person.
I don't know. It depends whether they came over for coffee and told you they were nervous all the time.
Well, that's true. And maybe I'd want to have somebody to play chess with, and I might want an over-methylated person who's studied it.
Yeah. Bill, I can't tell you how much I've appreciated this conversation. It's been really good. We've gotten down to some very useful details that anybody can use, whether it's a psychiatrist or a person on the street. You've really said a lot of really important things here, and I really appreciate it. What we're going to do at Core Brain Journal is we're going to give 3 of your books away, and it's going to be, after this is published, it's going to be 2 weeks from the time that this is published, and then we're going to cut it off. But we're going to do the books, and we're going to come in and have a drawing, and it's going to be a lot of fun because we want to encourage people to read what you've written. Now, we think it's really important, and we're going to do it. We're going to do it on this one, we're going to do it on the next one, and the one after that. So I'm going to encourage our listeners to tune in for the next one. We're going to really get into something that Bill talked a little bit about here, was pyrrole and copper. We hit it a little bit, but we're going to really dig down into those because they're more they're not so much in the public eye as right. Methylation is. So we thought we'd start with kind of the easy part and then get down to where these difficulties and imbalances can create significant mental problems and perpetuate them if they're not addressed properly. So, Bill, with that, I think we're going to close up. Thank you so much again. I really appreciate you taking the time.
Well, you're perfectly welcome. I look forward to our next meeting.
Thanks for listening to Core Brain Journal. We're working every day behind the scenes to bring you reports that connect research benches with those street trenches. Here we share the complexity of mind science because, as you know, details really do matter. One of the most pervasive, misunderstood challenges is how commonplace medications, like those written for ADHD, are used so regularly without clear guidelines. If you think you'd like more specifics, take a minute to download my 2-page PDF packed with video links and references on the absolute essentials of how to start ADHD medications. They're easily available at corebrainjournal.com/start. Thanks for listening. Do connect and stay tuned. Together we can make a difference.